Hello, good afternoon everyone. My name is Cristina Claver. I'm doing my PhD at ASTI, and today I want to present you our work called Assessing Accuracy and Completeness of GenBank for DNA metabarcoding towards a reliable marine fish reference database. Marine ecosystem monitoring is challenging with traditional methods, especially when we talk about the open ocean. So new technologies such as genetic approaches are becoming a better method to do so. Especially the last years, environmental DNA metabarcoding studies have exponentially increased. eDNA metabarcoding is applied to create species and community inventories, and the clear advantage of it is the capability for studying rare and elusive macrofauna. So, of course, limitations exist, and nowadays the major problem is the quality of reference libraries that are used to identify diversity. There are some requirements because they must be complete and accurate. If a species is missing in the library, it will never be detected in any study. So here we will present a case study of reference DNA status for European marine fishes, a commercial and economical important target. Regarding the reference database, we selected GenBank, the largest public DNA reference database, which is used worldwide. And our work started creating a fish biodiversity checklist to be later used in the sequence retrieval. So we gathered information from FishBase, the main fish information source online, literature, and surveys adjacent to this project. To ensure trackability of information, synonyms and misspellings were checked in the World Register of Marine Species and were included in next steps to avoid loss of information. So once the checklist was completed, we selected the most used markers for fish eDNA metabarcoding, which are the cytochrome C oxidase subunit 1 or COI, 12 and 16S ribosomal RNAs, and cytochrome B genes. So we retrieved from GenBank all the mitochondrial sequences available for our species according to their description name. With this information, we performed a gap analysis for the genes, and within COI and 12S regions, we selected the most common barcodes, the universal Fulmer-Leray and the Myfish Antelio for 12S, and performed a pairwise distance analysis of the sequences. And this allowed us to evaluate the performance of the dataset and detect doubtful sequences. The obtained checklist included 1,966 marine species. The biggest group was the Actinopterygii, then Elasmobranchi, and a few species of Holocephali, Petromyzontia, and Myxini. About 46, 8, and 27,000 sequences were retrieved from the chosen genes. The COI region showed the highest species coverage, about 70%, and cytochrome B the lowest, 47%. 12S and 16S had similar values. And the large number of sequences of cytochrome B gene, despite its low species coverage, is due to a high number of records corresponding to a small number of species. So this clearly means that a lot of sequences don't mean a lot of coverage. So now focusing on the barcodes, the COI pair showed more than 60% of species coverage, with a small proportion of species for which the gene sequence itself is available, but the barcode region is not. And comparing to COI, 12S barcodes were available for a smaller number of species. And among them, Teleo covered a larger number than Myfish, about 250 more species. In the next step, we selected the barcode regions and performed an all against all alignment to check the accuracy of these markers. So we calculated the distance matrix for each barcode, and we obtained distance values among species. The intraspecific represented with SP, and between species of different taxonomic levels, the interspecific distances that were split according to taxonomy, GE, same genus, FA, same family, etc. So we expect that the distance between sequences of the same species is small or null, and as species get more separated taxonomically, are also genetically more distant. Here, sequence similarity decreased as pair of sequences belonged to more distant taxonomic groups, but not always. For intraspecific distances, some pairs had significantly lower similarity scores, reaching up to 80% for the COI markers, and at the same time, some interspecific distances were very small or null when comparing species belonging to different families, orders, and even classes. It is not possible to set a threshold, but we established similarity levels for each barcode according to data distribution. So level 1 was established according to interspecific values, and for COI markers, the level 1 ranged from 100% similarity to 98.5% similarity. And for the 12S markers, instead, level 1 corresponded to total similarity, so it was 100%. We can translate box plot information into numbers. So this heat map helps to analyze the data structure. Regarding interspecific distances, we detect some pairs for all barcodes with a small or null divergence that correspond to distant groups, reaching up to 100% similarity. For MyFish and Teleo, the level 1 means 100% similarity. And also, among species comparisons, we observe some pairs that have bigger distances than expected. Less than 90% similarity for the Fulmer Lerai and up to 92% for Teleo. So this data can include erroneous records, but it can also be suggesting that big variability can occur within the same species. GenBank provides global information and records are uploaded all around the world. So there are some species, such as the lightfish, the Maurelicus moyeri, which has a global distribution, and some authors claim that this species should be split into 13 species. On the opposite side, there might be species that have already been split into different groups, but are genetically close. So it makes sense that the species of the same genus are close. And for example, we can see that for Teleo, there are 16,000 comparisons at family level within the same family that are identical. There are more records than at genus level. So if we filter our data and extract these sequences, we can check that 95 of them correspond to interactions within Gadidae family, and mainly to the Atlantic cod, Gadus morua, and the polar cod, Boreogadus saida. More than 10,000 comparisons among these 16. So once we reach this point, interpretation is a tedious work. One way to face it is by performing phylogenetic trees and these kind of approaches. Here we choose a network like plotting of these unexpected results, as you can see at the right side, and we can visualize some taxonomically distant species records that are more similar than expected. I cannot go into them all in this presentation, but for example, in blue dots we can see at order level 3 marlin sequences that match completely with the swordfish sequence. Reaching also up to class level, in gray, we see one chromy sequence that is identical to a thorny tintelfish sequence, the Gramincolepis brachioculus. So these seem to be erroneous sequences, as they are not the only ones that we have from this species, and they do not make this kind of matches. So, in brief, we can conclude that the most used fish metabarcoding marker lacks records of half of the European fish species. In this context, we highly recommend checking occurrence and availability of species sequences in our study area with information of surveys and literature. In some cases, intraspecific variability is wider and interspecific distances are narrower than expected, and different reasons are possible. We are currently working on a semi-automatic pipeline to identify when does this happen and to facilitate their removal if needed. So thank you all for your attention, and now I will happily answer all your questions.